So it's the end of my time at the Putney School, and for my final project, I wanted to look into something related to climate change and renewable fuel sources. Looking through a number of different potential topics, I came across the bacteria Clostridium thermophilum and the potential ways you can optimize its chemical pathways to make ethanol from plants. When I say make ethanol from plants, what I really mean is how do you convert cellulose, the stuff plants are made out of, into chemical ethanol, or C2H5OH. Now, ethanol is an alcohol, and one common way you can synthesize these is through fermenting a sugar. Cellulose is actually a polysaccharide made out of glucose monomers, which is just a fancy way of saying that it's a bunch of glucose molecules, or sugars, strung together. So before getting into Clostridium thermophilum and what makes it so special, let's first look at the traditional way that you might be able to turn cellulose into ethanol. So first, you would need to get your cellulose. The most common available source of cellulose in the creation of ethanol is actually lignocellulose. Lignocellulose is what's known as a complex of lignin, or the stuff that gives plants their woody texture, and other cellulose components. Some examples of this might be things like wood pulp or corn stalks. In order to get access to the cellulose, which again is what we're looking for, we need to break apart the complex. This can be done in a variety of different ways, but the end product is typically a watery slurry that contains separated cellulose, lignin, and sometimes hemicellulose. But let's just forget about that last one for a little bit. Alright, so you have your watery paste. The next thing that needs to happen is to break apart the cellulose. You can see from this picture that the cellulose is a chain of molecules which can be separated out into individual glucose molecules using water and what's known as a cellulase enzyme. A cellulase enzyme allows H2O molecules to bond with specific points in the cellulose structure, breaking it apart in a process known as hydrolysis. Hydro, meaning water, and lysis, coming from the Greek word lysis, or loosening. So with the help of these enzymes, we're almost ready to begin the fermentation process. I'm going to gloss over this a bit, because it involves a large number of smaller steps that I'm not all that familiar with yet. But basically, glycolysis is the next thing that happens. Glucose molecules are converted into pyruvate, which allows for the production of cellular energy, or ATP, which is used in the processing of more glucose. There are a number of different ways this can happen, which I will go into a bit more later. But generally, a number of enzymes and cellular structures help convert the glucose into glucose 6-phosphates, then to fructose 6-phosphates, and then into pyruvate. I might put a picture on the screen or a link in the description that explains this step. Now we're finally ready to begin fermenting alcohol. So again, there are a number of different ways this can happen, but generally they involve more enzymes and more shifting of ATP. But eventually, you can ferment a number of different products, including ethanol. From there, it's only a matter of removing the excess waste, like water and lignin, from the beginning of this process, and then you have pure ethanol that you can use as fuel or for drinking. So in theory, this process would allow us to create unlimited ethanol from plants as inputs, but in actuality, it's a bit more complex. The first thing is that all enzymes in cellular structures are expensive. Now, this may not be expensive in the sense of money, but the cost of consistently adding enzymes and bacteria to different parts of the system can be very resource intensive. The next problem is that this is a very multi-layered system, with lots of different bacteria and enzymes being added into it. This complexity allows for many different inefficiencies to occur, which can reduce the overall yield and viability of this entire operation. So this is where bacteria like Clostridium thermophilum become useful. Clostridium thermophilum comes from a genus of pathogens and bacteria, but some species have the ability to make useful products from inputs, or in the case of C. thermophilum, ethanol from cellulose. What separates C. thermophilum from other industrial methods of creating ethanol is a property known as compact bioprocessing, or CBP. CBP is when multiple biological events happen in a single reactor, or in this case, multiple steps of our cellulase fermentation can happen as a result of a single bacterium. Scientists realized that C. thermophilum was able to naturally produce what are known as cellulosomes, or packages of cellulase enzymes, as well as provide a pathway for the creation of pyruvate and ethanol. Because all of these operations were contained within a single organism, it had the potential to be much more efficient. But if Clostridium thermophilum was so much better at creating ethanol than all the previous industrial methods, why is it not more common now? One major issue with C. thermophilum is that it contains multiple pathways in its fermentation process. This can result in different ways that glucose 6-phosphates are synthesized, but more importantly, can result in random end products being produced. One of the main pathways that pyruvate takes once fermented is the creation of acetate, which decreases ethanol yield. But if it produces other products which make it an ineffective ethanol producer, why does this even matter? It's important to realize that compact bioprocessing and the modification of microorganisms is still a very emerging field. Clostridium thermophilum's wild type may not be very good at doing one thing, but with further study, scientists have been able to remove genes which code for production of other products, allowing for greater ethanol yields. This metabolic engineering allowed one strain of Clostridium thermophilum ALK2, to reach 90% of the theoretical maximum ethanol yield. Advancements such as this, and research into other potentially viable CBP organisms, could be the future in the creation of renewable biofuels. So, while we are just starting to understand the potential for using modification and study of more efficient CBP organisms, we are still a little ways off from creating a truly efficient organism. Maybe once we do, we'll be able to figure out a way to close the loop on climate change. Biomass is one of the largest types of carbon sink that exists, and maybe we can find ways to grow and convert this directly into ethanol or some other kind of fuel. This is an interesting topic, and I think I'll look into it more even after this project ends.